Hello, everyone. Welcome to join today's webinar. I'm Iris, Digital Marketing Supervisor at GenScript, and I'll be the moderator for today's webinar. A little background about GenScript for those who are new to us. GenScript is the leading life science research and application service and product provider in various fields, from basic life science research to translational biomedical development, industrial synthetic products, and cell therapeutic solutions. To learn more, download the resources and click the link below to visit genscript.com. In today's webinar, we have Dr. Gracie Cha, Field Application Scientist, to present on neoantigen peptide synthesis service for precision therapeutic discovery. Dr. Gracie Cha is a Field Application Scientist in GenScript Asia Pacific. She completed her PhD degree from Tokyo Institute Technology, Japan. She has 5 years of research experience in chemical biology and peptide engineering. Her previous research focuses on establishing functional biomaterial utilizing self-assembling peptide materials for 3D cancer cell culture and drug testing. Upon joining GenScript, Dr. Cha supports peptide synthesis platform services as the Field Application Scientist. She provides technical support to commercial team on pre and post sales activities. This webinar will take around 45 minutes, and we will have about 15 minutes for Q&A section. At any time during the section, type in your questions in the Q&A box down below, and we will answer them after the presentation. If we didn't get to answer your questions, we will email the answer back to you after this webinar. Also, we encourage you to submit your answer and to the survey on the left-hand side of your screen. Now, without further ado, let's welcome Dr. Cha. Dr. Cha, over to you.
Okay, thank you, Iris, for the kind introduction. So a very good afternoon to all. Thank you for taking your time to attend the GenScript neoantigen peptide synthesis service for precision therapeutic discovery. So today I'll cover 4 sections, and this includes the overview of neoantigen therapies, neoantigen peptide application. Then I'll move into GenScript's neoantigen peptide synthesis platform, and finally GenScript's peptide synthesis service, which covers RUO and RUOplus grade peptides. So what are neoantigens, and how does it impact the cancer immunology? So cancer is the world's leading cause of death, with accounting for nearly 10 million deaths yearly. So researchers have realized that the standard method treatments such as chemotherapy, radiation are not the best solution for cancer, because it still holds like around 56% of the mortality rate. And patients withhold standard treatment because of the negative side effects that they might suffer from. And the off-the-shelf therapeutic shows very limited efficacy for cancer with high tumor mutation burden. This is because the development of the therapeutic method is much slower than how a cancer can mutate. So in recent years, cancer immunology therapies is catching the attention of researchers. So gene mutation caused by genetic instability during the carcinogenesis always occur in the non-coding region and coding region, and the amino acid sequence changes caused by the mutation in the coding region can produce the protein that are not found in normal cells. So this protein can actually activate the immune system and lead to the immune system's attack on cancer cells. So immunotherapy is generally divided into 2 categories: active and passive immunotherapy. So active immunotherapy refers to eliminating cancer cells by stimulating the body's immune system, while passive immunotherapies refers to the passive acceptance by an organism of antibodies, cytokines, and transformed immune cells that can directly act on the tumor. So tumor vaccines includes cell vaccines, DNA vaccines, mRNA vaccines, polypeptide vaccines can actually recognize the protein that presents on specific cancer cell, arrest the cancer cell growth and prevent cancer recurrence, and finally clear the cancer cell that remains after treatments. So, what are neoantigens? Immunotherapies for cancer treatments have been around for more than 10 years. However, the current treatments only targets on the shared targets such as oncogenes, tumor-associated antigens, or immune checkpoint. So the immune checkpoints are the receptors on the surface of the T cell which has been bound by their ligands and are able to inhibit the T cell-based immune response against the target cells. So many cancer cells are able to avoid immune detection, such as the expression of the PD-L1, which is the ligand for the PD-1, which will bind T cell checkpoints and inhibit the immune attack. So checkpoint inhibitors are proteins which block specific immune checkpoints, such as PD-1, and initiate T cell-mediated immune response against tumor through the binding of the T cell receptor and the tumor-associated antigens. So clinical trials using all these checkpoint inhibitors have shown great potential, although the effect is limited in many cases, especially in solid tumors where the checkpoint inhibitors cannot make direct contact with the tumor microenvironment. So due to the mutation-rich nature of the cancer cell genome, cancer cells often produce proteins that are incomplete or flawed. So those truncated and flawed proteins are what we term neoantigens and can act as a patient-specific targets for novel immunotherapies. So since every patient's mutation lot is different and every tumor mutation lot is different, so the targeting these mutant proteins can actually has a more focused attack on the only cell in the body where the mutant's protein is expressed. So by directing the immune system to attack cells that express the neoantigen, off-target effects are maximized and focused destruction of tumor cells is also maximized. So what exactly is precision medicine? In precision medicine, individual patients' tumors are biopsied and used for NGS whole exome sequencing, RNA sequencing, or proteome identification. Through the bioinformatics analysis of this sequence compared to the healthy tissue, so researchers can identify the treatment which will work for the specific tumor biomarkers present in the patient's tumor. There are two types of precision medicine. So the first is to match the patient's tumor biomarker to the database for clinical and therapeutic known to be effective for those specific biomarkers. So second are the personalized therapeutics which are designed from scratch to target on the patient tumor-specific antigen known as neoantigen, where this is where we will focus on. So there's reason why personalized neoantigen are the preferred target. Because normally neoantigens are not expressed at a very high level to be able to detect by the immune system, because they are not recognized as self-antigen. And then once a neoantigen forms, there is a very low chances of it to mutate again and leading to the therapeutic antigenic escape. So neoantigen therapeutics tend to target multiple neoantigens simultaneously, avoiding possibility of immune escape. And each neoantigen can be matched to specific TCRs, and therefore, their immune response can be closely analyzed in vitro before being used in a possible therapeutics. So neoantigens are expressed as peptide within the cancer tissue from single amino acid deletion or insertion because of how they are generated, and they tend to be highly hydrophobic in nature and having a very strong tendency to aggregate. However, since they are minimally affected by antigen escape or weak immunogenicity, they make a very great target for immunotherapy. And in terms of personalized cancer immunotherapy, neoantigens are used as the primary target to initiate their immune response against a patient's tumor. So this personalized treatment usually begins by sequencing the patient's tumor and then comparing it to the database. So from there, clinicians will use bioinformatics tools to identify the mutanome, which is basically a peptidome comprising of the patient's specific tumor mutation, which are not common amongst the tumor types or healthy tissue. Because these targets are so unique, and therefore they can perform a very strong immune response. While the other types are the adoptive T cell transfer, so the steps are very similar to the personalized cancer vaccine, but a large library of neoantigen peptides are needed in order to identify the patient's specific T cell with high affinity and immunogenicity against the tumor neoantigen. And once the strong TCRs has been identified, it is necessary to expand the specific T cell or the genetically engineered patient T cell with high affinity TCR, and then finally infuse back into the patient in order to initiate the tumor killing. So the World Health Organization has estimated the number of the new cancer cases reported across the globe to rise for 70% over the next 20 years. So neoantigen targeted therapies will be a continuously and have a high demand, and continuous effort is needed. So let's have a look on the neoantigen peptides and its application. So no matter what types of neoantigen targeted therapeutics, researchers will require the production of synthetic neoantigen peptide for the application, particularly on the personalized cancer vaccine, precision mRNA vaccine, and personalized solid tumor immunotherapy. So in the standard stages of neoantigen therapeutic development, so researchers will require the production of neoantigen peptides on the stage 3 to 5, which are the peptide production, preclinical screening, and preclinical testing. So neoantigen peptides are required for preclinical functionality screening of bioinformatics candidates. So for the T cell therapy, these peptides will be utilized for TCR binding affinity and immunogenicity analysis, and the neoantigen candidate for the PCV development will be used for T cell stimulation assay. After the new therapeutic has been developed, the peptide will be tested on animal for the preclinical testing, mainly for the toxicity of the therapeutics, such as the adjuvant and other types of delivery vehicles, such as the nanoparticles. So in all these stages, neoantigen peptide will be used to confirm the immune responses initiated. So in the standard stages of neoantigen therapeutics development, researchers will require the production like I mentioned before, on stage 3 to 5. So, but neoantigen peptides are actually very difficult in synthesis and also purification. So neoantigen peptides will require all different types of synthesis in order to produce a very high purity peptide at certain required use. Specifically, neoantigen peptide can be difficult to synthesize because of like 5 types of characteristics, including length, charge, hydrophobicity, yield, and purity. So for length, neoantigen tends to be 9 to 11 amino acids, representing the specific epitope, or they will be larger than 25 amino acids in order to be processed or presented by antigen-presenting cell onto MHC. But peptides over 20 amino acids tends to be very difficult to synthesize. And usually we require a combination of between microwave and solid phase peptide synthesis. While for peptide charge, because neoantigens are only expressed in cancer, so they tend to contain a very high charges modifying amino acid, which are not seen within the natural tumor-associated antigen. So many synthetic steps only works with specific isoelectric point, and therefore, like further from neutral a peptide is, the synthesis will be more difficult. So for the third one, Because neoantigens are produced from random amino acid throughout the peptide, so hydrophobic side chain are bound to the introduced within the sequence. So this hydrophobic amino acid will lead to a stronger chance of aggregation and therefore limit the solubility in water, which in turn will make the purification very difficult, and it's very difficult to obtain a very high yield. So neoantigen peptide synthesis can also be difficult because each application within the various stages of therapeutic development requires a completely different yield, mass, purity, and percentage of the target peptide within a sample. So because of the flexibility required, it's difficult to find a reliable provider which is able to accommodate yield and ranging from a very large microscale library from milligram up to gram level in one individual peptide. Because of this, so GenScript has tailored its neoantigen peptide service in order to make this type of hydrophobic peptide at the quality and purity that you need. So let's have a look on the GenScript's neoantigen peptide synthesis platform. So what can GenScript provide where there are so many challenges in each step? So during the stages of the neoantigen precision immunotherapy, you might face the challenge such as the prediction of the peptide sequence, multiple mutation site, patients with urgent needs, so you need to obtain your neoantigen peptides in a fast mode, and finally the commercialization at the end of the process. So what can GenScript provide? So there are 4 steps involved in our peptide synthesis platform. So in order to accomplish this type of difficult neoantigen peptide synthesis, GenScript has developed our own neoantigen peptide synthesis platform utilizing the reliable technology and tools to achieve at least a 95% of success rate in synthesis, particularly on the difficult neoantigen peptide. So in the first step, every neoantigen peptide sequence that comes through our order system will be analyzed by our NeoPre-bioinformatics algorithm. So in order to determine the synthetic difficulty, and the algorithm can actually provide the information to choose the best method to synthesize each peptide based on its predicted difficulties. So in step 2, the second stage of our platform, so those neoantigen peptides will be synthesized according to the recommendation of our NeoPre algorithm, and our peptide expert will be using either one of the methods such as microwave, liquid phase, or solid phase technology, or under the combination of either 2 of these technology to obtain and maximize the success rate of the neoantigen peptide synthesis. So neoantigen peptide that has been synthesized using our NeoPre and synthetic technology, they will go through a very strict quality control based on the individual customers and application needs. So all GenScript peptides undergoes a total quality management testing, including the mass spec analysis, HPLC analysis, and also throughout all this process will be carried out throughout the peptide synthesis and purification process under our quality control service. So in addition to the standard quality control test, so GenScript also provides other types of quality control services as listed here. So this including our endotoxin analysis and removal services. So endotoxin are lipopolysaccharides, which are the major components of the cell wall of the gram-negative bacteria, which can stimulate some unwanted immune reaction in B cell, macrophage, or stimulate the T cell expansion even at a very low concentration that might ruin your whole experiment. So to enhance and make sure that the experimental results are trustable, so GenScript now offers the customized peptide with guaranteed endotoxin control service to reduce the endotoxin level up to 10 EU per milligram, which is very ideal for most of the cellular assay. Besides that, we also provide guaranteed TFA measurement and removal because the residue of the TFA during the peptide cleavage can actually cause some effects on your subsequent assay and data. So in final step, after you obtain the peptide, we also will provides the design of like 7 different types of peptide library in order to narrow down the perfect neoantigen sequence. So the most popular peptide library design service for neoantigen peptides are alanine scanning, overlapping and T-cell truncation library. So in an alanine scanning library, so each amino acid will be replaced with an alanine. So in turn, this will provides the end user to determine which is the most important individual amino acid to the peptide function. And different from alanine scanning library, so the overlapping library will replace the individual amino acid by simply deleting the amino acid over a certain threshold. Because of this, it's great to identify where the neoantigen epitope is within the entire neoantigen peptide sequence. And finally, the T-cell truncation library are similar to overlapping library, except that you can decide on the multiple epitopes length to put into one pool in order to filter out the perfect length of the neoantigen or T-cell receptor. So at GenScript, we constantly enhancing our capabilities in order to produce the highest quality reagent possible for the community. So in terms of neoantigens, we currently focus on both research use only grade peptide, RUO, or RUOplus for functional screening, animal studies, and post-injection efficacy analysis. For clinical testing, we are still suggesting the GMP grade neoantigen peptide because it will apply directly to the patients. So let's have a look on the differences between the RUO and RUO plus grade peptide, which is needed in the whole process. So for RUO grade peptide, currently our capacity includes the production of 12,000 peptides per month with more than 98% synthesis success rate. So we are able to provide the peptide as fast as 5 days up to like 200 amino acid with a purity between 70 to 98%. And all the peptides have to undergo strict QC and QA tests before we release it. And currently we can even synthesize up to kilogram level for RUO grade peptide. So what actually supports our high capacity in peptide synthesis is because we have a very strong core in-house technology, such as the PepPower, which integrates both liquid and solid phase peptide synthesis with the microwave technologies. So this can actually contribute in high synthesis success rate of over 98%. Besides that, we also have our PepHTS library platform, which can produce a very large scale peptide in fully automated platform. So this platform can support the synthesis of up to 600 peptides at the same time. While for the large-scale peptide synthesis platform, we have our self-developed high-throughput synthesizer, which can support up to the capacity of 2 kilograms per month. So GenScript provides a full spectrum of high-quality peptide service, ranging from the custom peptide synthesis to the high-throughput peptide library and peptide array service. So powered by our PepPower platform. So we have the professional chemists, which make us become the first vendor that can deliver peptide in as fast as 5 days at guaranteed 100% guaranteed quantity. So our services are mainly focused on standard peptide synthesis and express peptide synthesis, as well as standard peptide library and microscale peptide library. So for our custom peptide synthesis service, you may choose between the regular, fast, or rush peptide synthesis with only differences in the sequence length of synthesis, purity, and also type of modification as listed in the table here. So basically for the rush peptide, there's only over 30 types of modification to choose from, while for the regular peptide synthesis, you can actually choose up to over 300 types of modification, depending on your need. Yeah. So let's have a look on the peptide library. So to meet different purity demands for the multiple application, we also developed like 2 types of the peptide library, including the standard peptide library for milligram level and microscale peptide library at a reduced price, which is ideal for the preliminary screening and the proteomic application. So basically, peptide library will be needed when you need to apply for the T-cell immunotherapy, vaccine development, cancer research, antibody and T-cell epitope mapping, proteomics, and other biological assays. So there are more than 300 types of modification to choose from, and for the detailed list, you may actually browse through our website for a complete list of modification. These are just some of the examples of the modification provided and also based on the different types of application needs. So what I want to emphasize here is that all GenScript's peptide undergoes a total quality management testing, include mass spectrometry, HPLC, throughout the peptide synthesis and purification process. And currently, we also include the solubility test for every peptide synthesis. And there's other types of QC testing, which are upon request with certain charges applied, but you can always select the QC testing which fits your need. So in vaccine development, we are actually suggesting the RUOplus grade peptide. So let's have a look on the differences between the RUO grade and the RUOplus grade peptide. For RUOplus grade peptide, it is suitable for neoantigen peptide synthesis because it provides the full traceability of key material with dedicated equipment and line clearance to avoid the cross-contamination. So the peptide will be synthesized in a clean room production with certified workshop by authorized third party and operated by our own scientists. So they will provide the batch production record to make sure the reproducibility and also test record for customers' reference. The filing service are provided upon request, and this is very important if you are intended to go for IND application. So for RUOplus peptide synthesis service, it's a guaranteed higher quality because it's a new quality assurance system to will be applied for these peptide services because it includes the key material management, line clearance, and environmental control. And we provide the documentation system to establish a separate document management system. And then it, like I mentioned before, it's produced in a C-grade clean room, which is certified by authorized third party to provide the environmental assurance, microbiology, and endotoxin control. So we have also dedicated equipment which separate the purification chromatographic column, freeze drying, vialing, packaging for each project, which is validated cleaning procedure for the equipments. So we also do the testing like equipment validation and method customization based on customers' needs. And you can also customize the QC service that you need. So currently our peptide service for neoantigen can provide up to the length of 50 amino acid with purity up to more than 98%. And the modification are upon your request. So the capacity that we are having now is using the multi-channel microwave synthesizer, which can produce the 24 peptides in a single batch. The amount will be ranging from milligram to 10 grams. The standard QC will be applied on each peptide, including mass spec, HPLC, and solubility test. So like I mentioned before, the QC can be customized. So the more important one will be endotoxin and TFA control. And the delivery method format will be in either lyophilized form or solution form upon request. So if you are worried about the solubility of the peptide, then you can request for us to help you to prepare it in a solution form. And we also helps in the free vialing limited up to 5 tubes. And we also have the batch record and experimental record for your needs. And the overall turnaround time is 2 to 4 weeks. So the special need will be adjusted appropriately and the IQR is required. Just to emphasize a bit for the PCV development for the first one, so you might have a crude peptide library for the in vitro screening purposes. And then for the preclinical PCV development for non-human primate, actually you can start to try using the RUOplus peptide. While for the phase 3, researcher might require the vaccine and non-vaccine control peptide for clinical in vitro efficacy screening. While for TCR affinity and immunogenicity screening, for the phase 1, crude peptide library is a good choice for the in vitro screening. And on the second phase, you might choose for the overlapping library to map the specific interaction with the epitopes. While for the phase 3, you can choose for the alanine screening to determine the most important amino acid for the therapeutic efficacy. So just to share a little bit with you on the case study that GenScript helping with. So for the case 1, we are actually helping with the mRNA vaccine for the high throughput screening. So this is our R&D grade peptide screening. So we actually helps to provide 600 sequence of around 15 amino acid within 24 hours time. This is because we have our in-house platform that supports PepHTS library platform. It's multi-channel automated industrial production, and this is why we can synthesize the peptide efficiently with high yield. So another case is the PCV. So as I mentioned before, PCV is a very difficult peptide synthesis, and when it requires fast delivery because patients actually cannot wait. So during this case, we are required to synthesize the neoantigen peptide for PCV development in a fast mode. The neoantigen peptides were synthesized in clean room with our NeoPre algorithm, PepPower technology, as well as our high-sync technology, which utilize and optimize reaction temperature, and also condensation reagent to actually increase the coupling efficiency. This is how we managed to provide a very fast service with high-quality peptide just within 7 days. So the final step will be, because currently we are collaborating with the third party to help with commercialization until IND approval. So we are also in the preparation to launch our own fully GMP neoantigen service in order to provide the personalized immunotherapy community with a more reliable provider for this type of difficult neoantigen peptide. So we also having a professional team standby to assist you with the neoantigen immunotherapy design, as well as the transfer process from the RUOplus grade to GMP. So we are planning to continue our highly technical approach to peptide synthesis in order to ensure that we are a truly trustworthy neoantigen peptide production team. So currently, GenScript actually contributed for over 256 organizations in their needs for neoantigen peptide synthesis with success rate of 95%. So many of the research utilizing our neoantigen peptides were published in international journal with great testimonials. So this is a little bit of the overall summary of our neoantigen service, where we actually will be able to help in basically all your neoantigen needs with customized service with flexible application that matching the economical and fast delivery mode. Yeah. So that's all from me. Thank you.
Thank you, Dr. Jia, for the great presentation. For all the attendees online right now, feel free to drop your questions in the Q&A box, and we'll be able to answer it. We have a few questions from the flow now, so let's get started. The first question is, what are the advantages of GenScript's neoantigen peptides?
Okay, thank you for the questions. So, like I mentioned before in my presentation just now, so neoantigen peptide usually vary in length, charge, and hydrophobicity. So it is very difficult in synthesis and purification. So GenScript has developed our NeoPre algorithm. So this type of algorithm can actually able to determine the peptide synthesis difficulty based on the sequence alone. So with this type of algorithm, we're able to synthesize neoantigen peptides with over like 98% of the success rate. And we also have a very great turnaround time of about less than 10 days. So once your peptide has been produced, GenScript also offers like other types of library design tools such as alanine screening, overlapping T-cell truncation to actually help you optimize any neoantigen sequence. So these are the advantages of using our neoantigen peptide, as we promise to provide you with the one-stop solution for all our peptide users.
Great, thank you, Dr. Jha. And the next question would be, what are the differences between neoantigen peptides and normal peptides in the manufacturing process?
Okay, GenScript actually offers high-sync technology specific for the neoantigen hydrophobic peptide production. So our high-sync technology actually utilizes an optimized reaction of different temperature and condensation reagents to increase the coupling efficiency. So by using this type of advanced coupling methods, so high-sync actually allows the synthesis and manufacture of the previously unproducible peptides. So apart from that, so like I mentioned, GenScript actually providing different grade of neoantigen peptides, such as for research use only, RUO, or the RUOplus peptide. So in terms of RUOplus peptide in the manufacturing process, so these peptides are produced in clean room with dedicated equipment. We have separated where they separate the purification, freeze drying, vialing, packaging process. So RUOplus peptide actually provides also with the documentation, which actually will help you in the process when you need to proceed up to GMP transfer and also commercialization.
Okay, thank you. Next question is, the neoantigen only focus for cancers or any other diseases?
Okay, for neoantigens, because for us, it's neoantigen is to target on the mutation that display only on the cancer. So basically, in this case, we are using the neoantigen peptides for cancer only. Yeah. Because neoantigen are actually the new and novel types of cancer antigen that presents on the cancer's tissue, but not expressed within the wild-type tissue. So this is why they are expressed within the individual tumors of a specific patient, rather than being shared with other patients that are suffering from even the same type of cancer. So they are actually very specific to the patient alone. Yeah, it's like custom made for each patient.
Okay, thank you. The next question is, did GenScript could provide peptide side chain modification or site selective modification method for the neoantigen?
So actually, every type, like I mentioned before, we do provide different types of modification for our peptide. Well, for the case of neoantigens, because we have the neoantigen peptide usually are shorter compared to other types of peptide. So if you intend to like do a very specific epitope mapping. So you can always like require us to do a specific modification for the epitope that you need on your neoantigen peptides. So actually all of these are doable. So what you need to do is actually you provide us with the peptide sequence and also like inform us and discuss with us on your downstream application and like what type of modification that you need. And certainly we will consult with our production team. So because we have the NeoPre algorithm, so all this can actually screen for your sequence, whether it is doable or not. Yeah. So yeah, we can do the modification.
Okay, thank you, Dr. Jia. Here's the next question goes to, are there any same neoantigens expressed in the patients, or they are always different in patients?
Okay, because neoantigen peptide, what we trying to do is the first step is we get the biopsy from the single patient. So usually every patient will definitely get certain different types of sequence. It might be similar, but it might not be the same. This is why we have to offer the library design tools such as like alanine scanning. So probably the sequence are of the same length, but there is just slightly difference, and we do not know like which amino acid is actually playing the important roles to provide the immune responses. This is where we need the alanine scanning or probably the overlapping scanning to make sure and optimize the neoantigen sequence. But I will say that all the neoantigen sequence are actually specific to each patient themselves. Yeah.
Okay, thank you. The next one is, what is the capacity of GenScript's neoantigen peptide production?
Okay, currently GenScript able to provide the RUO grade neoantigen peptides with length up to 50 amino acids. So this is because our multi-channel microwave synthesizer can produce like from milligram up to 10 gram scale. So we do like provides other types of QC, including the endotoxic control, TFA removal. But if you opt to try out with the research grade only for functional screening, animal studies, and also probably the post-injection efficacy analysis, we actually able to support up to like 12,000 peptides per month with more than 98% of the success rate in synthesis. So we are trying to like upgrade our synthetic methodologies and also our bioinformatic tools. So this can actually help us to increase our capacity. So if you like wish to know that our capacity exactly, so you can actually approach our team to customize your neoantigen peptides for your project. And I'm sure that we definitely will make sure to match our capacity with your need. Yeah.
Okay, thank you very much. The next one is, what is the difference between the antigenic peptide and neoantigen peptide?
Okay, for the neoantigen peptides, like I mentioned before, it's from the tumor itself, and then after that we send for the NGS sequencing. After the NGS sequencing, actually the researchers, they will actually compare with the database itself. So there's actually, in the system, there's actually, in the system, actually, we do have the database that contains all the sequence of compare with the healthy tissue. So from here, clinician actually can identify like which types of which sequence among from the sequence that they obtain from the NGS is actually that will work specifically for the tumor biomarker presence in the patient's tumor. So, yeah. Okay.
Right. Thank you so much. Maybe we'll move to the next question is that what is the next step for neoantigen peptide in precision immunotherapy?
Okay, so at GenScript, we actually constantly enhancing our capability to produce a higher quality peptide possible. So for in the production of like neoantigen to help in the precision immunotherapy, we actually currently advancing from not only providing RUO grade peptide, but also RUOplus grade peptide for the animal studies and also other functional screening. And currently, like I mentioned, we are in the preparation to launch our own fully GMP neoantigen service in order to provide a better personalized immunotherapy community with as a reliable provider of the difficult neoantigen peptides. So we also have a professional teams standby to assist you with your neoantigen immunotherapy design, as well as the transfer procedure for the RUOplus and GMP. As we are continuing to our highly technical approach to peptide synthesis, so we really want to ensure that we are truly a trustworthy neoantigen peptide production. Yeah, so hopefully we can provide more helps in the precision immunotherapy for all the researchers.
Yeah, sure. Thank you, Dr. Cha, for all the answers. And thank you for all the great questions. So I think we have completed all the questions from the floor. So if the attendees have any questions after the section, feel free to email us and we will back to you with the answer. So now we're about to end the webinar today. Again, thank you, Dr. Jha, for the great presentation. And to all of you who joined us today, and I hope you find this session beneficial to your work. And thank you for joining us again, and have a nice day.